My name's Kelsey Moody, and I'm the CEO of Ichor Therapeutics and our various portfolio companies. And today I'd like to take a little bit of time to talk about our protein engineering platform and how we've used it and how we plan to use it to discover and develop new classes of drugs for age-related disease. Now, before I jump into that, I would like to thank the team at Ichor that's been responsible for supporting and performing all of this research, with a particular shout out to our COO, Aaron Wolf, who's in the audience, and our CSO, Adam Blandon, who are the founding inventors of the technology that I'm going to be presenting today. Obviously, I'd also like to thank the various foundations, funds, and angel investors, again, many of whom are in the audience, who have continued to support our research across a variety of different platforms. Your continued support is greatly appreciated and really allows us to do what we do. So for those of you who follow us, you may remember that Ichor launched a drug discovery program called Intoxerine last year, which focused on cellular senescence, which has been a recurring topic at this conference today. And the basis of our technology is shown here. This is a solubility tag. We call it RP tag. And what this allows us to do is if we express a protein of interest and we attach this to the N-terminus, it allows us in a scalable way to make very difficult to express proteins. Now, we like being biophysics nerds, so no one appreciates this, but what this allows us to do is it's an enabling technology where we can do high throughput drug screening on very difficult to make targets that we believe are lead to much more biologically relevant assays. Now, I can't say a whole lot about the progress of our work, except to say that today we can announce publicly for the first time that we've closed a $10 million deal with Juvenescence to develop our collection of small molecule compounds targeting cellular senescence. That press release came out about 2 hours ago. But wait, there's more. That was only the first generation of the technology platform. And today what I'd really like to dive into is how we want to continue developing the technology and what our second and third generations of this tech look like. So in early 2017, we identified that we could actually split this solubility tag into two different fragments. We're very original, so we called it RP tag large for the large piece and RP tag small for the small piece. And what's significant about this is that these two tags bind each other with antibody level affinity. So the graph on the left, the y-axis, shows fraction bound from 0 to 100%, and we titrated in additional protein on the x-axis. And if you look at the black line, what that's showing is that RP tag large and small bind each other, again, with antibody level affinity with a KD of about 1 nanomole. This compares very favorably to our control antibody, which was an anti-His antibody and its epitope 6-His tag. And we also see that when we combine RP tag large or small with BSA, we don't really see off-target binding. It seems to be pretty specific for its target. Now, what this allowed us to do is generate a whole suite of protein chemistry tools for our company. We could mix and match the RP tag large and small with plates, chips, fluorophores, proteins, beads, you name it. And that was able to drive a lot of the R&D at our company. These products are starting to begin being released through our website, through a Ichor portfolio company, Recombipure. The kits are online at recombipure.com, and we're going to continue to release additional kits using this technology later on this year. Now, as our automation and protein engineering teams were wrapping up our early discovery work with Intoxerine, we started thinking about what program we wanted to deploy them on next, because they like to be in the lab 14 hours a day, so we've got to keep them busy. And as we were looking through the various options of what we wanted to deploy them on, we made an interesting observation that I think is worth stating here explicitly. Where we see pathology in the body is often not where the pathology originates. If we think about cancer, only 10% of deaths from cancer are driven by the primary tumor. Something like 90% are actually metastases that are in different areas of the body. If we think about atherosclerosis, right, the liver is what's manufacturing 80% of our daily supply of cholesterol, and yet the location where we see the lesions are throughout the vasculature and the heart. And even when we look at seemingly straightforward diseases like Parkinson's disease, we think about Parkinson's disease as the loss of dopaminergic neurons in the substantia nigra driven by Lewy body formation and alpha-synuclein. And yet it's just started to be believed and appreciated that Parkinson's disease does not originate in the brain. Rather, the enteric nervous system of the gastrointestinal tract, so the nerves in your gut, overproduce misfolded alpha-synuclein, which migrates by way of the vagus nerve into the brain, and that's actually where the disease originates. I was floored when I found this out. I thought that was incredibly interesting, and that led me to look a little bit more into how the gut is involved in age-related processes. And as we looked into this further, what we recognize is that the gut is actually an overlooked and undervalued frontier in longevity science. And if we had a technology that could modulate the microflora of the gut, this could have a profound application on the way we manage and treat various age-related diseases. Now, the challenge of targeting the gut, of course, is that biologics are very susceptible to the harsh environments of the GI tract. Indeed, the entire point of the GI tract is to break down biologics so that your body can absorb them. But the RP-TAG system actually comes from a deep-sea geyser thermophile, so we were wondering if it might be robust enough to survive utility in the GI tract. And it turns out it can. So the graph on the left shows heat stability of the RP-TAG system. So what we did is we took anti-His antibody or the RP-TAG large and repeat boiled it over and over and over again. Now, as anyone that's done bench work in the audience might appreciate, antibodies don't enjoy being boiled very much and completely lost activity within 2 or 3 boiling trials. But the RP-TAG large, we were able to repeat boil 26 times with only about a 50% loss of activity. I will tell to this audience that our COO actually convinced me to spend a few $100s on antibodies to throw in the autoclave as a control to see if we could autoclave this thing. Apparently, antibodies don't enjoy being autoclaved. RP-TAG survived, granted with reduced binding affinity, but it did survive and was still bioactive after a full autoclave cycle. An additional feature is its pH stability. So typically biologics have a very narrow operating range for pH. And yet when we did a binding assay, which is shown in the middle graph, where we took RP-TAG large and bound it to 3 different versions of the RP-TAG small, one that was the native, shown in black, one that was optimized for tighter binding, shown in red, and one that had fast kinetics and was optimized for kinetics, shown in blue, we see a very wide operating range for proper folding and binding from pH of 4 until about 10 or so. Now, the RP-TAG natively is a ribose binding protein, and I don't have the data here, unfortunately, but when we supplemented this with ribose, we actually expanded the pH range from 2 till about 10, which is pretty incredible, again, for a biologic. Additionally, we wanted to know if the RP-TAG was able to survive enzymatic degradation, so we compared its ability to survive pepsin treatment at gastric pH, which is shown in the right panel. Bovine serum albumin, our control, was degraded at very low concentrations of pepsin, and yet RP-TAG large was able to survive at orders of magnitude higher concentrations of enzyme. So this led us to ask, could this protein survive the gastrointestinal tract, and could we use this as a therapeutic modality? Now, this is a bit of a busy slide, but bear with me because the nuances are important. So what we did for this experiment is we had RP-tag large, which is the protein. It is able to bind RP-tag small on the C-terminus over here. In our formulation, we added ribose to stabilize it. And it is known that you can conjugate biologics to vitamin B12 such that they can be absorbed through intrinsic factor pathway. So we engineered a cysteine with a free thiol and bound vitamin B12 to it as a delivery system. Now, people have tried using B12 delivery of biologics for a while. The problem is the cargo gets destroyed in the gastrointestinal tract. We added on a 3X flag tag here so that we could detect it. And we appreciated that enterokinases are produced by the duodenum, and that would be able to cleave this. So we also engineered in a proline to make it resistant to enterokinase degradation. And we fed this to mice at different time points over 24 hours, and then we pulled serum. And the way that we read it is traditional ELISA, where the capture was RP-tag small, RP-tag large bound to it, and then we used an anti-flag tag antibody in order to detect it. By structuring the readout in this way, the only way that we could get signal is if we had full-size bioactive protein, because we're capturing it on one end and the readout is on the other end. Our serum PK after gavage feeding is shown here. So we are indeed able to create an RP tag that is able to become orally bioavailable at reasonably high concentrations for a first pass, which is rather unheard of for biologics. Now, of course, who really cares if we can bind RP-tag small? This isn't a clinically relevant target. So we then set out to establish proof of concept that we could bind a clinically relevant epitope. Now, unfortunately, I don't have time to go into the protein engineering of how we did that here, but suffice to say we did successfully accomplish that, and we engineered RP-tag large to bind platelet-derived growth factor beta. And those results were published in ACS Biochemistry a couple months ago. So collectively, what we have is a platform technology that in theory could replace conventional antibody therapy with a product that is 1/1000th the cost to manufacture, could potentially be eaten instead of injected, is stable indefinitely at room temperature. It has an unfolding half-life measured in decades, and can target previously inaccessible areas of the body, like the GI tract, where antibodies cannot function. We've created a new portfolio company, which we're calling Octus Biologics, to develop this platform. And we're preparing to initiate a high-throughput mutagenicity screen to engineer RP-tag large to bind epitopes of every single antibody in phase 3 trials, every single FDA-approved monoclonal antibody, as well as a number of high-priority gastrointestinal and immunosenescent targets. Now, as many of you in the audience know, Ichor Therapeutics only focuses on diseases of aging, and we work diligently and collaboratively to really try to deliver on the promise of this field. I understand that there's a lot of scientists in this audience, so if there are any groups who we can help with any part of this technology platform and portfolio, please get in touch with my team. There's several of us running around here, or some member of my staff or me during the conference to talk to us about how we can assist with your projects. And with that, I thank you for your attention.